All right. So, in what we have just been talking about, you were learning about DNA, right? DNA is in our nucleus of our cells. It contains all of the genetic information that makes us who we are. So lots and lots and lots of information is stored in that DNA. We learned it's made of two strands that have a bunch of letters, A's, T's, G's, and C's. And the order that those letters come in, that's our genes, right? And so if we compared each of us, most of the letters are the same, but there's some differences. And those differences are what makes each of us unique and different from each other. So DNA has all of the information to make you or to make any other organism. RNA is another similar type of molecule to DNA, but it has some different uses and some different functions, and that's what we're going to be talking about. So RNA sounds like DNA. It's also a nucleic acid. That's what the N and the A is. But it's got some differences. So RNA is, again, another large molecule, a large polymer, but it's only single-stranded. It doesn't have two sides to it. An RNA molecule only has one side. It's made of really pretty much the same things. RNA has a phosphate group. It has a sugar. In this case, it's a different sugar. It's called just ribose. Remember, DNA was deoxyribose. So there's a phosphate and a sugar, and it has bases. It has some of the same bases. It has a C for cytosine. It has a G for guanine. It has an A for adenine. But here's the difference. It doesn't have the T. It has a different base there. Instead of the T for thymine, RNA has a U. It's a different base called the uracil. So that's a difference between DNA and RNA. Okay. Now, RNA has a different job to do. We'll go through this several times. We're going to watch a couple of videos and so forth. So there's three different types of RNA. So usually we put a letter in front of them that tells which one we're talking about. mRNA is messenger RNA. tRNA is transfer RNA. And then rRNA is ribosomal RNA. And basically all of these different types of RNA have a special job to do to build proteins. And they're all working together to build a protein, because that's what our genes do. They tell our body how to make and when to make and how much to make of these certain proteins. So messenger RNA is the molecule that carries the instructions, the recipe, you might think, from the DNA out to the cytoplasm. The ribosome, the rRNA, kind of coordinates the building of the protein, and then transfer RNA brings in the right amino acids. So here's a little comparison between DNA and RNA. DNA, two strands, two sides. RNA, one strand. The sugar is different. In DNA, the sugar is deoxyribose. In RNA, it's just called ribose. The bases. DNA has A, G, T, and C. RNA has A, G, C, and U. They're both nucleotides, and they both are made of the same basic components, a sugar, a phosphate, and then the base. Okay, so how does this all work? That's what we're going to be talking about. So really, our genetic code, the G's and C's and T's and A's that make up all of our DNA, what that really is, it's information about how to make proteins. All the proteins that form our whole body, the instructions to build those proteins are stored in our DNA. We have about 3 billion letters You know, the activity you were doing, how many letters were you working with? How many did you put in a row? You remember, Isaiah? Just 8. And even with just 8, it's pretty complicated. There's lots of different sequences that you can put those letters in. Imagine with 3 billion letters. Just the different varieties are almost infinite. So the way that this code that's in our DNA is actually used has a few different steps that we'll talk about. So first off, the DNA has basically like the master recipe about how to build the. Let's use an example. Let's use insulin. We know insulin's a hormone. It's a protein. It's really important for how our body works. So somewhere in our genetic code, somewhere in those 3 billion bases, is the instructions about how to build insulin. Okay, so let's say our cells are going to build some insulin. What happens? First off, the DNA inside of the nucleus stays there. It's kind of stuck. But that's where the recipe is, right? That's where the instructions to build insulin are stored there. So the first step to build insulin or any other protein is this instructions in the DNA are copied over to what's called messenger RNA. That messenger RNA just has a copy of the instructions. It's a smaller molecule because that's all it has is instructions to build insulin. So it can get out of the nucleus and it goes out into the cytoplasm of the cell and it attaches to a ribosome. Remember, ribosome's job is to help build proteins. And now it has the instructions. Has the instruction about how to build insulin. And it's at the ribosome. To build insulin, you need a whole bunch of hundreds or thousands of amino acids to get linked together. And which amino acids are put together in which order that's what the instructions are that are in the messenger RNA. And so what the last mRNA, transfer RNA, does is starts bringing in the correct amino acids. Starts bringing them to the ribosome, they start getting attached, one to the other, until the whole insulin molecule is made. Thousands of these amino acids, and they form insulin. And then it can be released, it can go into our blood and do its thing. That's how the instructions in DNA are actually used to make an actual protein. We can look at some diagrams of it. The first step is called transcription. This is the part when the DNA instructions get copied to messenger RNA, transcription. And basically what happens in this process, it happens in the nucleus. The DNA kind of partially unzips. And the recipe is copied from DNA to RNA. And we see that here. So here's the double helix of the DNA. Here's a section that's sort of unzipped. And an enzyme called RNA polymerase is being used to create this messenger RNA that has a copy of the DNA. So if the DNA letters were G, G, C, A, T, A, C, C, G, A, T, the messenger RNA is going to match up. What's going to be here?
C, C, G,
now,
T, T,
hold
A,
on.
T,
What did I just say about RNA here? It doesn't have a T. What's it have instead? A U. U, U, A, U, G, G, C, U, A. So it's very similar to what you were just doing in that gizmo lab or in the game, but there's that difference. RNA doesn't have T's. It has a U instead for uracil. So this is transcription. The instructions about how to build insulin are here in the DNA. It's copied. The instructions are now on this messenger RNA molecule, which can go out of the nucleus, can go out to the ribosome in order to start building a protein. That process of taking the recipe, the instructions, and using it to build a protein is called translation. It's using the coded messenger RNA to actually build the protein. Every 3 bases, every 3 letters in the messenger RNA is called a codon. It's a letter. It's a code for one of the amino acids. Transfer RNA brings in the correct amino acid because it matches up. So here's our messenger RNA, GUC, CAG, and so forth. Every 3 letters is a code for one amino acid. The transfer RNA matches up, it brings the amino acids, and they start putting together the amino acids in the correct order. There's a special codes that are called start, which start the protein being made, and when it gets to the end codon, that means the protein is finished. You can use a chart to figure out what messenger RNA codons are a code for which amino acids. If you remember back to proteins, there's about 20 different amino acids. So the chart allows you to figure out which is a code for which. So let me show you how this chart works. Maybe I can annotate. Yeah. All right. So let's say the first codon is ACU. What I do is I look up the first letter, A. There it is. So it's going to be in this row somewhere. Then I go up to the top. The second letter is C. Where do they meet? Right here in this box. And then I look at this side, follow that across. Here we do. We have ACU. ACU is a code for this amino acid therine, THR. Okay, let's look at a different example. What if it's GAC? GAC. If I follow where those all connect, here it is. GAC. That's the code for this other amino acid. Let me give you one. CGA. Which is that a code for? CGA.
What do you mean? Like a...
So which of these
ARG.
Yep. Yeah, these things, these letters here, those are the amino acids. So what is it?
ARG.
What did I just tell you? I forgot what I even said. CGA, CGA. Yep, there it is. Yeah, that's a code for arginine. So you don't have to memorize the chart, obviously, but you should know how to use it. And so that's one. So arginine is one of the amino acids, and glycine is another one. And so these amino acids are getting put on one after another to form the protein. Just another picture of it. We see in the nucleus, the DNA gene copied to messenger RNA, which goes out to the ribosome. Transfer RNA molecules start bringing in the correct amino acids. And these green dots, it looks like a pearl necklace or something. That's the protein that's being built based on the recipe that comes from the messenger RNA, which came from the DNA. Uh-oh, that's not what I wanted. Just another view of it. These are all sort of showing the same thing. And because I know it's a complicated process, I just want to keep reinforcing it. So this is a good one because it gives you the steps in order. 1, DNA in the nucleus has the instructions, the template, it's sometimes called. It's copied over to messenger RNA. That's what we see here. The messenger RNA can squeak through the nucleus and go to this red thing, which is the ribosome. The ribosome kind of reads the instructions, and the transfer RNAs start bringing in all of the correct amino acids to form the protein. So again, transcription, and then translation. So transcription, the DNA is copied to the messenger RNA, goes out to the ribosome. These green transfer RNAs are bringing in each amino acid one after another, hundreds or thousands of times until the protein has been created. Okay.